Okay, hello everyone. Thank you for being here. My name is Sara Oukkal. I'm a second-year PhD student in Lyon, France, and I'm going to talk to you about domestication of endogenous viral elements in tachinid flies, asking whether it's an evolutionary convergence with parasitoid wasps. So first, let me introduce my super cool model, the parasitoids. They are organisms, usually wasps, spending their immature stages attached to or within a host, like a caterpillar, for example, ultimately killing it while emerging. There are two types of these, the ectoparasitoids, which develop on the surface of their host, and the endoparasitoids, which develop inside the host. And because they develop inside the host, they are highly confronted to the host immune system. And these endoparasitoid wasps, they had many solutions to counter the host immune system, but one of the most interesting ways involves a third player in the host-parasitoid relationship, and it's viruses. So some endoparasitoid wasps, they acquire genes from viruses, and they use them to form particles they inject to the host while laying eggs, and these particles, they will inhibit the host immune system, thus increase the survival rate of the progeny in the host. These acquired genes, acquired from viruses, they are integrated to the host, to the parasitoid genome. And because of that, we call them endogenous viral elements, for short, EVEs. And the fact that they are used by the wasp for its own sake is called viral domestication. These particles, they are not in all endoparasitoid Hymenoptera. They are indeed like 5 well-documented cases of integration of viruses that you can see in red. And the fact that they are independent integrations means that they are kind of evolutionary convergence amongst endoparasitoid Hymenoptera to face the challenges these parasitoids have with their hosts. And because of that, a former PhD student in my team, Benjamin Guinet, conducted a study on many genomes of free-living ectoparasitoid and endoparasitoid Hymenoptera, and he concluded that there are more endogenous and domesticated viral elements in endoparasitoids compared to the two other groups, which suggests a kind of association between the symbiosis with viruses and this endoparasitoid lifestyle. But the question here is what happens outside the Hymenoptera order. Because there are endoparasitoids beyond the Hymenoptera order, and these may also be associated with viruses. And that's what I'm asking here by working on Tachinidae, tachinid flies. They're a family of Diptera, the biggest group of endoparasitoids outside the Hymenopteran order, which means they are highly confronted to the host immune system as well. I'm asking 2 questions here. The first one is, are there endogenous viral elements in Tachinidae genomes? And the second one, how does it compare with species with either the same lifestyle and different group, like endoparasitoid Hymenoptera, or the same group but a different lifestyle, like free-living Diptera? For the first question, I assembled 38 Tachinidae genomes from short-read Illumina reads. And these genomes were assembled initially for the sake of the Horizon project, which aims to unravel the link between horizontal transfers and the ecological relationship between a host and its parasitoid. And if you're interested in this kind of stuff, go and see my colleagues Audrey Portal talk today at 5:15 in the evolutionary ecology session. On top of these genomes, I added 15 Tachinidae genome of really high quality from the Darwin Tree of Life project. And what is interesting about this data set is that before my PhD, there were no Tachinidae genomes available at all, and now we have 53 ready to address many other questions on them. For the second question on how does it compare with species with same lifestyle, different group, I added 38 Hymenoptera endoparasitoids analyzed by Benjamin Guinet. And for the same group, different lifestyle comparison, I added 25 free-living Diptera that I assembled from publicly available reads. So let's jump into the methods. The first thing I wanted was to detect EVEs in my genomes. Without going really into details, the first thing was to detect homology between my insect genome and a viral protein database. Once I had these candidates, I created clusters of homology with candidates and viral proteins, so like groups of proteins that look alike. And after that, I wanted to check whether my candidates were indeed endogenized, because especially in short-read Illumina assemblies, we also have reads from the microbiome of the insect in the assembly. So I did not want to have free viruses and think of them as being integrated to the genome. So I checked whether the scaffold containing the candidate either had sequencing depth similar to the insect scaffolds or contained busco-eukaryotic genes or transposable elements on it. So by doing that, I found eaves in all the species that are analyzed at variable rates, as you can see here. I had an average of 32 eaves and a maximum of 219. But what we noticed pretty quickly was that we had a significant difference between higher quality genomes that you see in blue here and lower quality ones in green. And this is because our detection of eaves relies a lot on the presence of eukaryotic genes on the same scaffold as the candidate, and therefore the higher the quality of the genomes, the longer the scaffolds, the better the detection. So for this question of finding eaves in tachinid flies, I took all these genomes, but when I compared tachinid flies to other groups, I only took into account the green ones because the other groups had only Illumina short-read genomes in them, so I didn't want to create a bias. So when I compared the Tachinidae family to Hymenoptera endoparasitoid and Diptera free-living, I saw that there was a higher variability in the number of eaves in Hymenoptera, but in the end, no significant differences between tachinid flies and the two other groups. When I grouped eaves by viral families, I found out that I had a wide variety of families, both RNA and DNA ones, and most of them were known to infect Diptera, or at least insects in a more general way, and these are the ones in bold on the plot, which means that the content of genomes in eaves is quite a good proxy for viral infections in general. We also had two families that were not known to infect Diptera, so maybe it's the case, but we should keep in mind that here we are seeing the closest relatives to our eaves in the databases, but the initial donor of these sequences could be a different unknown virus. So when I compared these viral families between groups with the same lifestyle, I found some families that were shared between Tachinidae and Hymenoptera, but not all, and especially some major families with a lot of eaves were not shared between the two groups. But when I compared Tachinidae with free-living Diptera, I had a majority of viral families that were shared, and all major families with a lot of eaves were shared. So viral families, their proportions are more linked to the taxonomic group than the lifestyle here. So zooming out, we are regrouping here eaves by genomic structure, and by genomic structure, I mean double-stranded, single-stranded, DNA, RNA viruses. So the DNA ones are in blue, and the reddish ones are RNA ones. And what we see here, that we have a majority of double-stranded DNA viruses amongst eaves. And when we compare it to what we find in databases in terms of viruses infecting insects, we see the trend is completely reversed. In the databases, we have a majority of RNA viruses. And this overrepresentation of double-stranded DNA viruses amongst eaves may be because in insects, the viruses, especially the RNA viruses, do not often have a DNA form, unlike retroviruses and vertebrates, which means that here, in this case, the viruses that are the most prone to integrate are the double-stranded DNA ones, because they have a structure that is similar to the structure of the insect genome, and they replicate in the nucleus. So, jumping back into the methods. So now that I have my eaves, what I wanted is to reconstitute the integration events, which means that I have a phylogeny, and I want to know when these genes came into these genomes and if they are shared or not. For that, I started by doing phylogeny. I did phylogeny for the species, but I also did phylogenies for each cluster containing candidates and viral proteins. After that, I regrouped the eaves, the ones that were homologous if they formed a monophyletic group in the species tree, and the ones that were not homologous by distance on the scaffold if they were close, the genomic structure, and the species profile if I found them in the same species. After that, a step that is ongoing, not done yet, is to detect whether these eaves are domesticated or not, and for that I'm performing a DNDS analysis, concluding on purifying selection if the metric is below 1. So here I have my integration events mapped on the phylogeny. And as you can see, most eaves are specific to one species, and these are the blue dots you see on the end of branches of the phylogeny. But I have 51 viral genes that are shared by at least 2 species, and these are the ones you see in green. The fact that they are shared by several species means that they integrated into the common ancestor of this species, and their persistence in genomes could suggest a possible domestication. And I will check that with the DNDS analysis results coming soon. So if I go more into the function of these genes, I tried to check whether I had eaves that could be related to functions, could help the parasitoid in its challenges. So I found genes that were probably involved in the production of viral particles, genes that could inhibit the host immune system, and some genes that could be related in general to parasitoid functions. And to finish, I wanted to highlight a really interesting case of near complete integration of a Hydrosa virus. Hydrosa viruses are double-stranded DNA ones in my species here. So these viruses do not integrate into the host genome during their natural cycle. So which means that here, if they're integrated, it means that they got accidentally through non-homologous end joining recombination or through the machinery of transposable elements, for example. So here on the plot, you have all the genes from the virus, and the green ones are the ones I found shared by several species, and the blue ones were the ones I found in at least one species. So to conclude on that, the first question was, are there eaves in Tachynidae genomes? And the answer is yes, there are. And there's another representation of double-stranded DNA viruses with mainly Hydrosa viruses. And there are a lot of eaves that could potentially be useful for the parasitoids by their function, and they may be domesticated. And for the second question, how does it compare to species with the same lifestyle, same taxonomy group? All groups showed similar abundance in eaves, but Tachynidae had more in common with free-living Diptera in terms of viral diversity. So thank you all for your attention. I would like to thank the conference for inviting me here and my team for helping me on the everyday basis from the Laboratory of Biometry and Evolutionary Biology in Lyon, France. And I'm looking for a postdoc for January 2026, so if you want to talk about that, we can link there. Thank you.
Thank you all so much for coming. I'm Zoey DeLaney, and I'll be presenting on monarchs and their parasite. I've been working on this research for the past 2 years, so this is all very hot off the press, and I'm excited to share our new results with you. So first of all, my lab focuses on the sexual selection of monarch butterflies, which is actually quite unique for Lepidoptera, as instead of pheromones and mating dances, the males have actually evolved to use forced copulation with females. I, in particular, was interested in lock and key dynamics and how this might influence that mating behavior. So lock and key dynamics refer to how well the male genitalia fits inside the female genitalia, and it can be very important for understanding species that are in a sexual arms race. For monarchs, this indicates how sharp and needle-like the male Adagus has become, and in response, how the female Ductu bursae has become hard and sclerotized. So even though lock and key dynamics are very important, there's actually very little still known about monarch genitalia, and so I began my research by just teaching myself how to dissect it out. When I started, I focused heavily on the very tip of the male Adagus because I wanted to see if a certain shape or size influenced mating success, and a majority of them appeared like this, nice and clean, but then I started to see some that appeared dirty. And I thought this might be a one-off until it happened again and again and again. And so I went back and looked at the females and found the same thing happening. So again, this is just the female Ductu bursae, where the Adagus actually is inserted. And a majority were clean, but I kept seeing these dirty genitalia popping up. So I finally took this to my PI. And his jaw hit the floor. He was like, Zoey, this is not dirt. These are actually the spores of a parasite that we're not supposed to have in the lab, known as Ophiocystis electroscira. So what is Ophiocystis electrosclera, which I'll now be referring to as OE? So OE is a specialized, widespread protozoan parasite that has detrimental effects on monarchs, including deformed wings, lower flight endurance, low reproductive fitness, and often death. Adults with OE can easily be identified through a traditional method known as taping, where a piece of tape is placed on their abdomens, and when it's removed, it takes a sample of scales with it. When we look at this under the microscope, if the adult is infected, we can see these distinct ovals that are actually the OE spores. So since OE is so virulent, it's thought to be one of the main drivers of monarch decline, which has been consistent and well documented over the past 30 years. However, if we look at the prevalence of OE, it actually varies quite wildly throughout migration, reaching its peaks from 50 to 70% in the middle of migration, and then at the ends of migration in Canada and Mexico at 2 and 5%. This is due to migratory culling, where sick individuals are unable to complete migration and they drop out of the population. What's puzzling here is how these populations can go from such low prevalences to such high prevalences so quickly. One of the reasons we think is that OE has several known routes of transmission, the first being vertical from parent to offspring, when the female spreads spores through ovipositing. The second is environmental transmission, where an unrelated adult spreads spores on a plant, which has an egg or a caterpillar on it. Then there is horizontal transmission, which occurs between two adults in direct physical contact, and this mode is the only one that does not cause pathology. Our data suggests that there is possibly an additional cryptic route of transmission, sexual in nature. To assess this, we did 2 experiments, the first being to determine if OE is transmitted internally between copulating adults, and the second is to determine if there are any adults that have internal OE on their genitalia, but no external OE on their abdomens. So for both experiments, I had the same methods, where we began with inoculating caterpillars with OE by giving them a leaf with OE spores on them to eat. After about a month, they would eclose as adults, and we would then tape their abdomens using the method I described earlier. And this is an actual picture of the tape, so it just looks like an imprint of their abdomen. When we look at that under the microscope, we can assess their external OE infections. And then after monarch death, we would remove their abdomens and place it in KOH for about 12 to 24 hours. And when we were ready to dissect out the genitalia, and looking at that under the microscope, we could assess their internal OE infection. So for the first experiment, the sexual transmission of OE, we had 50 cages that all had 2 uninfected males and 1 heavily infected female. The red dots here represent the OE spores on her body. And so after the first mating, or if there was no mating, after 10 days, the 2 males were removed, retaped, and dissected to assess their internal and external OE scores. What we expected to see was that both of the males would acquire some level of external OE infection from proximity to the female, but that only the mated male would acquire an internal OE infection. So here we can see on the x-axis were the unmated and mated males, and the sample sizes aren't exactly the same due to not all the cages having a mating occur. And then on the y-axis we have our parasite score, which is a log scale from 0 to 5, 0 being no spores, 1 being about 10, and 5 being over 100,000. What we saw from the first experiment was that externally, almost all of the males contracted OE, but that the mated males contracted a significantly higher amount. We then dissected all the individuals to assess their internal OE, and we saw that all of the mated males contracted OE on their genitalia, and that almost none of the unmated males did. We did have 2 of the unmated males contract about 10 spores, but this could have been from an attempted mating that we weren't aware of. And these were significant, which suggests that it is sexually transmitted. And so just for reference, this is the typical amount of sexually transmitted OE, which is a score of about 2, which means that all of those dots make up about 100 spores. So for the second experiment, which was the cryptic OE infections, we began with inoculating about 200 caterpillars. And then once they closed, we taped all of them, and all the dots are the individuals along that same scale from 0 to 5. Here we also labeled the scale with 0s being clean, and the 1 through 3s, or the lower level infections, are considered contaminated as they don't experience such severe effects from the parasite. And then 4 and 5 are often referred to as infected or heavily infected because they experience the most severe results. And as you can see, our experimental inoculations produced a majority of 5s and clean individuals. We then dissected them all to assess their internal OE infections, and we saw a correlation where all of the heavily infected individuals had OE internally, although it varied in severity, and that a majority of the clean individuals did not have any internally on their genital. This is a level 5 Aedegus, and all of those little clumps and dots are just tens of thousands of OE spores, just to keep that in mind. And so how does this compare to the wild? Well, there's a study done that showed that actually less than 10% of wild monarchs are these heavily infected 4s and 5s, and that there's 25% of those monarchs are contaminated individuals. How can this be? Well, these heavily infected individuals are usually so riddled with parasites that they can't perform the 3 Fs: flying, feeding, and of course, fornicating. And so without a lab assistant to feed them, they're dying off in the wild, and they're not actually contributing to any transmission of OE. So this is much more accurately representative of wild OE prevalences and intensities. These are the scariest points on this figure. These individuals here had 0 OE spores on their abdomens, but internally they had OE spores on their genitalia. This could mean that about 9% of monarchs harbor these cryptic infections where they can look and appear healthy, but they're actually able to sexually transmit OE. To put that into some perspective, if we go back to that puzzling question from earlier, and we look at those 2 and 5% that researchers are confused how those are reaching 50, 60, 70%, and we factor in that some of those individuals could be cryptically infected, it would make a lot more sense how they reach these high prevalences quicker. So overall, over the last 2 years, my lab has discovered OE spores in the genitalia tracts of monarchs, and our data suggests that this is readily transmitted through copulation. We've also found that there are likely cryptically infected individuals in the wild that can look and appear healthy, but are actually sexually transmitting this parasite. And because of this, we could be vastly underestimating the levels of infection and spread of OE among wild monarchs. In the future, we want to focus more heavily on offspring transmission rates and specifically determine the relationship between internal OE and vertical transmission. So again, this is a female ductu bursae where the egg comes out of, and if you just imagine an egg sliding out of there and picking up all of these spores along the way, and we've preliminarily been able to see this with some spores on the egg capsule. And this is important because the first thing a caterpillar does when it hatches is it eats its egg capsule. And if there are spores on it, it's going to become infected. So the next big question is how critical is internal OE for vertical transmission to be successful? So I'll keep you posted on that. And then I would like to acknowledge the Darude Lab at Emory for all of their help and resources that they shared, the FSNR grant at Davidson College for funding this research, and of course everybody in the VIA Lab for all of their hard work. I appreciate all of you very much.
Hi everyone, thanks for coming. My name is Monica Mowery, and I'm a new assistant professor at York College at the City University of New York. And today I'll be talking about testing co-invasion through population genomics of the invasive brown widow spider and its associated egg sac parasitoid. So we know that the world is facing significant rapid global changes, climate change, urbanization, increased pathogens, and extreme weather. And these changes have multiplicative effects. So one rapid change is the increased globalization and spread of invasive plants and animals. And we can use these invasive species as natural experiments to test ecological and evolutionary questions. How do these species spread around the world, and how might they spread associated pathogens, parasites, and parasitoids? So organisms can sometimes drift off course during migration with birds. Spiders can balloon, sometimes long-distance dispersal of hundreds of kilometers. But we know that with invasive species, most often humans inadvertently transport them to new places. So this is a map of global shipping routes in a year. And often species stow away in cargo ships or with produce and end up in new places where they sometimes successfully colonize, reproduce, and then spread past the point of introduction. And we can study these routes of invasion, points of origin, through population genomics. So this paper by Alex Blumenfeld found that the native range of termites in China was more genetically diverse than the invasive range in Japan and in the US. And they also found this bridgehead effect from this intermediate invasive population from Hawaii that was linking the native population in China to this newer invasive population in the southeast US. And in addition to studying one species invading, we can also use population genomics to look at these concurrent spread of hosts and symbionts or parasites, parasitoids. So this study on stink bugs found from the native range in East Asia that there was a congruent loss of host and symbiont genetic diversity during invasion. And in this other study on host aphid populations and an introduced parasitoid for biological control, they found high genetic differentiation in the host aphid, but low differentiation, high rates of gene flow in the parasitoid wasp population. So sometimes there are these congruent patterns, sometimes there are different patterns of diversity in the hosts and the parasitoid. So I'm looking at this in the brown widow spider. It's Latrodectus geometricus, same genus as the better-known black widow spider. They're likely native to South Africa, and they really thrive in urban and disturbed habitats, often hot and dry conditions. And they kind of unique among the widow spiders. They have this spiky egg sac, which through some experiments in the lab we found is an effective and a plastic defense against parasitoids. There are also genomic resources for this species. There's a recently sequenced reference genome. So the parasitoid that I'll be talking about is an egg sac parasitoid wasp, Philolemma latrodecti. It's a eurytomid wasp. And out of all the things we've seen in the field that attack widow spiders and their egg sacs, it's one of the major egg predators, egg sac parasitoids. So here's a female wasp sitting on a white widow spider egg sac. So the female lays eggs inside of the egg sac, then the wasps develop inside of the spider egg sac. And the wasp was first found in Hawaii in 1945, where there are actually no native widow spiders. So there was, in Hawaii, brown widows were first found in 1944 or so, and then the wasp was found in 1945. And then 10 years later in the Philippines, the brown widow was found 1 year, then the wasp was found the next year. So there's some anecdotal evidence that the brown widow and the wasp may have spread concurrently with this human-mediated introductions. So like I said, the wasp develops inside of the egg sac, and then after about 3 weeks, wasps emerge instead of spiders. So it has, there's complete mortality of any egg sacs that are parasitized. And in some sites, there are up to 50% parasitism of egg sacs. So it can be really damaging to widow spider populations. And for reference, here's a spider egg here on the right and a parasitoid wasp egg on the left. And the parasitoid wasps can crawl around. They're winged, but they appear to have limited dispersal in the field, and they seem to use spider silk to find hosts. And they have preferences for certain species over others. So my research questions are, how does genetic diversity vary in populations of brown widow spiders and egg sac parasitoids? Do the most diverse regions correspond to these likely native ranges of the spider, likely in South Africa? And then do patterns of genetic diversity correspond between the host and the parasitoid? So there are different historic scenarios. There's some uncertainty because we're not completely sure of the native range of the spider, and we don't know the native range of the wasp. But one hypothesis is that the brown widow and the parasitoid P. latrodecti share the same native range, where we'd expect higher genetic diversity across the same ancestral range, and then lower genetic diversity in the invasive range. And another scenario could be that the parasitoid wasp existed as part of the native fauna around the world, and then the spider, which we have dated invasions for, spread more recently. So I collected brown widows across the US, which is one invasion front. So brown widows were first introduced in the 1930s in southern Florida, and then they spread west, where they were more recently found in California. So you'll notice, in the US, there are 4,000 kilometers between the east and west coasts, so pretty large geographic scales between populations. And we have a sample of parasitoid, egg sac parasitoid wasps from 2 locations in Florida. Although parasitoids are also found in Georgia and Texas, but despite heavy sampling of egg sacs in California, they haven't been found in California. Brown widows are also in the Middle East, in Israel, Palestine, Jordan, Egypt. So here I'll be talking about the Israeli invasion front, where brown widows were first introduced in 1980 in Tel Aviv. And then they spread south to the Negev desert, where they were first detected in 2000. So we have a range of populations from Haifa all the way to Tel Aviv, the Negev, and then Eilat, which is on the Red Sea. So it could suggest a potential different route of invasion from a port city on the Red Sea versus a port city on the Mediterranean. And this is a typical brown widow habitat, kids' playgrounds and fences, and a lot of kind of disturbed habitats. So we also collected egg sac parasitoids in Israel. We collected from Haifa to Eilat, but only found the parasitoids in the Negev desert from about Beersheba to Mitzpe Ramon. And wasps, I found them in 2018. So brown widows, like I said, the brown widows first reached the Negev in 2000. And interestingly, in the 90s, Michal Sagoli and Yael Lubin collected hundreds of native Negev widow spider egg sacs, so white widows and brown widow egg sacs, and they did not find a single wasp. But the wasp appeared sometime after 2000. Okay, and I also collected brown widow spiders in 7 populations in South Africa, so about 1,500 kilometers here. So the Israeli populations are also on a smaller scale, about 400 kilometers between Haifa and Eilat. And then we'll have 2 populations of egg sac parasitoids from French Polynesia and Tahiti. So for the 2 species, so brown widows and then the P. latrodecti, I created DD RADseq data sets with 17 localities for brown widows and then 11 localities for the egg sac parasitoid. And filtered the DD RADseq data sets. So we mapped the brown widow DD RADseq data onto the reference genome and then used de novo assembly for the parasitoid wasp data. So I'll start with the brown widow spider. This is showing pairwise FST across all of the brown widow spider populations. So you can see this cluster here. These are the Israeli, Mediterranean, and Negev populations. There's relatively low genetic differentiation across these populations. There are also some geographic clusters. South Africa, the Cape Town region, there's low genetic differentiation here. Also, the northeast populations in Gauteng province in South Africa. But there is some connectivity, it looks like, between South African and US populations, whereas the Israeli populations here form their own cluster and are more distinct from the South African and the US populations. So you can see this clustering here. Interestingly, Texas here is kind of off on its own, apart from the other US populations, whereas the Mediterranean and Negev Israeli populations cluster here. So with the principal coordinate analysis of allelic diversity, you can see that there's a lot of overlap between the US and South African populations, and the Israeli populations cluster over here. With the parasitoid wasp, we see clustering, so low differentiation across the Israeli populations, but the Israeli populations are set apart from the US and the Tahiti populations. And there's some gene flow here. So the Tahiti populations are somewhat differentiated, and then the US, the two Florida populations, have relatively high gene flow. So you can see this clustering here of the Israeli populations versus the Tahiti and US populations. And similarly, you can see the Israeli populations in the principal coordinate analysis versus the US and Tahiti populations. So overall, in the invasive spiders, we found this clustering of the Negev and Mediterranean Israeli populations, whereas the Eilat spiders on the Red Sea were highly differentiated. This could suggest this potential different route of invasion. And in another study, I found that the Eilat spiders also had a unique divergent endosymbiont strain of Rhabdochlamydia. So more evidence of differentiation. In parasitoid wasps, there is the high distinction between the Israeli populations and the US and Tahiti populations, likely limited host specialization within the widow spider species as well, across brown widows, white widows, and black widows. And we didn't have a lot of populations where we had both the spider host and the parasitoids, but we found high genetic connectivity across large geographic distances and high heterozygosity in the invasive range. So what's next? More sampling in different populations. We see potential multiple introductions in the US, especially with these large geographic scales, and potential spread with other widow species. And right now I'm looking on historic specimens of the parasitoid to look at what was the genetic diversity in the 1940s and 50s with the parasitoids collected there. So if this research sounds interesting, please get in touch. I'm just starting up my new lab at CUNY. I'm happy to hear from you. And I'd like to thank folks at Ben-Gurion University, Aparna Lajmi from the University of Haifa, Sanbi, Tembile Kosan, Robin Lyle, and Jerome Marie in Tahiti. So thanks to everyone. Happy to take any questions.
Okay. Hi, everybody. I'm Nick Priest. And I have to say, every time I give a talk in a host parasite session, I always feel I have to apologize, because the systems we've seen so far are so much cooler than the damn fruit fly. But let me tell you this, that a question came up earlier about how do monarch butterflies deal with sexually transmitted infections. Is that different than for a larger dose infection? Okay, now that's a question we can address in the fly. And I think what I want to do in this talk is show you how you can, still with Drosophila, you can address fundamental questions, and hopefully those will have some use in, frankly, more interesting systems. So first, let me introduce the people that are actually doing the work. In particular, a lot of the work we're talking about today is from Jing Bo's PhD. And I also want to highlight Ali Jarvis, who's here. So what we're trying to do with Drosophila is bring our experiments to the public. And actually, we've now done, we've had interactions with something like 1,200 separate participants collecting something like 5,000 pieces of data that we're trying to incorporate into kind of citizen science where we bring the lab experiments to the public. So watch this space. And so we've run a whole series of these public engagement events, and that's sort of a snapshot of what it looks like. So I think behavioral ecology really is one of these topics that resonates well with the public. But it's not just for the public. We can also address long-standing fundamental questions, particularly about life history constraints. Where are the trade-offs? But also we can address questions about molecular biology. So if you think about it, there's a problem with our understanding of immunity. Because we think about it as a system designed to improve survival in infected organisms. But where's the reproduction in that? And don't you need reproduction for evolution? And so the immune paradigm actually needs a behavioral ecology perspective. And then we can in the behavioral ecology context, we can start to ask more interesting questions about what are the physical constraints that are in life history evolution and in evolution in general. So nutrition and temperature might alter those kind of things. So just a little background here. Of course, this is probably 101 here. When we look for genetic correlations, when we look, for example, between longevity and reproduction, a positive genetic correlation between those would actually be a pattern that would accelerate evolution. Individuals that live longer and produce more babies, those strains would tend to evolve faster. But a negative correlation would impede evolution, make it very difficult for the evolution of longevity or reproduction. Whereas when there's no correlation, those traits can evolve independently, right? This is 101. Now this is the life history prediction for that's supposed to be the fundamental to all of biology, that you can't live long and reproduce highly. Yet, when we look in nature, we're really struggling to find these negative genetic correlations between key life history traits. So is life history really constrained? This is a problem. Now, some people say, well, yeah, actually, maybe this is just a question about variation in nutrient acquisition, but there's also it could be costs of immunity that are constraining seeing this fundamental negative correlation that we're expecting. So in particular, there's a major problem with immunity research in that it's typically done with infections that have no bearing on what the organisms face in nature when they're injected with large quantities of microbes. And I mean, the standard immune paradigm, that they won the Nobel Prize for finding the innate immune pathways in Drosophila. But I would argue that maybe these pathways aren't even related to the types of infections that animals experience in nature. And that's because they kill the animal so quickly. So the assays are essentially, does the presence of a gene give the fly another 4 hours of life? Again, no reproduction in that at all. I've circled one gene in particular, DIF. It's a canonical part of the antifungal pathway. And so we basically use that to kind of test this question. And what we moved to was thinking about, hold on, maybe we should be focusing on topical infections and particularly low-dose sexually transmitted infections. And particularly STIs, because we think that about 80% of the known infections in insects are known to be sexually transmitted, and that's through physical contact during sex. So when we do the knockdown experiments, where I should have pointed this out, that flask there, that's how we infect the fly, the little symbol up in the far right corner. We dump the flies in a flask that have spores in the bottom and just stir them around. That's the main way we give them a topical infection. So it's very light touch. And they get STIs you do that to males, and then the females get to mate with them. So when we do the knockdown experiment, we see what would be the typical immune perspective, that if you have a functional expression of DIF, you live longer and it turns out you're more fertile. And if you don't have it, then it's a bad situation. So this would be like, yeah, obviously the immune system's adaptive. But of course, that's for a large dose infection that's somewhat implausible in nature. And then when you look at the STI context, you see sort of an opposite trend here, that actually it's very constrained. They can't evolve high survival without sacrifice and reproduction. So if you get rid of the immune system, actually, under an STI, they start cranking out more babies. And this got us thinking, well, maybe we're missing something here with low-dose infections. So we went to make an even lower dose infection, which is we take a tungsten needle, run it through a lawn of fungi, and gently tap the thorax of the fly. And you could sort of see this in, I don't know if that can run across if you see that at all, but I can't see the screen either. So, but where that red arrow is, that's sort of what it looks like. It's not much of an infection here. And we can measure, we could do this repeatedly and show that actually we can reliably give doses of varying degrees. And then ask, okay, so what do the flies do when they get these really low dose infections? And it turns out they do something completely different than you'd expect for immunity. They crank out babies. In the case of an extremely low dose infection where they have only a single tap on the thorax, we find that the viability of the eggs significantly drops. So this is in this one tap. It's dropping. And then, but if we give them 3 taps, then we see a boost in egg viability. And also, if the flies get sexually transmitted infections, that's where the males are infected and they mate with the females, then again we see a boost in egg viability. And so it's egg, and it turns out this is egg viability, not egg number. So there really is something internally happening, a constraint on the success of the young born as eggs. And it turns out that this is sort of consistent with the terminal investment, because we find that the strains, the treatments where you see the decline in egg viability are correlated with a higher survival. And a large dose infection, they suffer a lot more in terms of their survival. So, but still, where are the trade-offs here? We can plot this. So these aren't genetic trade-offs. These are just looking at individuals. And if what we found is if we just take the lifetime egg viability of a fly and map that to its lifespan, there's this strong negative correlation that flies that are long-lived are the ones that had low egg viability, and flies that are short-lived tended to have high egg viability. And this is true in all the treatments except the large dose infection. So under the large dose infection, effectively, longevity can evolve independently from egg viability, but not, its evolution potentially is very constrained here. So then we wanted to ask, okay, so what are the mechanisms? So how do monarch butterflies fight low-dose sexually transmitted infections? Is it fundamentally different? So then we started thinking in the fly strategies for this. How could we find this? Well, of course, flies get STIs, but before they get the STI, they actually get a warning of the insult that's about to come, because there's this process of courtship. And in the song itself, what we discovered in a project in collaboration with Mike Ritchie is that they turn on 2 genes in the head of the fly even before they have sex, just in response to the call. And we were able to show that this seems like a really good case of immune anticipation. Before having sex, they're preparing themselves for the infection they're about to get. And these 2 genes are TOT-M and TOT-C. So in a paper about 10 years ago, we were able to show that, yeah, actually in TOT-M improves survival of flies with sexually transmitted infections. But we always had this result that, like, the TOT-C didn't show anything. But that got us thinking, maybe it's not a survival response, maybe it's a reproduction response. And when we then looked for the trade-offs, so you can see below, I include the diff ones I showed you before, but at the upper bars, that's the TOT-C. And effectively under the STI condition, there was no trade-offs whatsoever. In fact, there was no fitness consequence of even having an STI for the different genotypes. But then we looked more closely at the pattern of reproduction, and what we could see is that actually the TOT-C is facilitating a shift in reproductive output. So what we're basically getting is genetic control of a fecundity compensation response. So if they have functional expression of TOT-C, then they boost the reproductive output on the first day after they get infected. But if they don't have TOT-C, then that pattern doesn't show. So TOT-C confers fecundity compensation. And there is a slight negative decline in their reproductive output after that day. Whereas with DIF, we don't really see DIF shows no evidence of this fecundity compensation. So TOT-C, actually this TOT-C should be considered one of the first genes, or possibly the first gene identified for a non-immunological response. So, and with high-dose infections, just very quickly on this, maybe we've been thinking about it all wrong with how immune systems evolve. Getting back to these genetic correlations, I showed you these before, but actually there's another one. There's a paper by Kirkpatrick and Landy, which says that actually when you have cross-generational fitness correlations, you can get rapid evolution. And sure enough, this is what we find in flies, that if we infect mothers, then it's a positive genetic correlation in the bottom, but if both mothers and daughters are not infected, then it's a negative genetic correlation. And overall, what we find is that if both the mothers and the daughters are infected, then those daughters get a fitness advantage through a maternal effect reproductive strategy. And this will have the consequence of an immune gene, or we don't want to call it immune gene, a reproduction gene, really, reproductive response to infection being an adaptive response. So where are the trade-offs? They're there. That's it for me, folks. Reproductive strategy changes, and we're looking at these physical constraints. Thanks.
Hey everyone, I'm Matthew Waller. I'm a PhD student from the University of Utah in the Clayton Bush Lab, and today I'm going to be talking about one of the chapters in my dissertation. So the parasite I study, it's a genus of flies called Philornis. They're parasitic nest flies, and they're a little bit different than a lot of the parasites we've heard today because the adult flies are not parasitic. It's only in the larval stage that these flies are parasitic, and they feed on the blood and tissue of nestling birds and breeding adults while they're in the nest. And Philornis is found across the neotropics, so it's in the Caribbean, South America, Central America, and Southern North America. And where I work in the Galapagos, I work on Philornis downsii, and it's invasive. It's a recent invasion without a long shared evolutionary history. Now, hosts vary in their ability to defend against parasites, and I'm going to go over a few groups of hosts that I think about a lot in my research. So the first group are vulnerable hosts. These are hosts that have low fitness, low survival, and reproduction when they're infested with parasites. There's also tolerant hosts. These are hosts that recover fitness by compensating for the damage done by parasitism. And because they can compensate for this damage, they can often support large numbers of parasites. A third category that I think about a lot that can overlap with these other two are reservoir hosts. And these are just groups of hosts that harbor parasites and ultimately end up transmitting them to vulnerable hosts. And these can overlap. It's pretty clear to see how tolerance could make a reservoir worse. And so if we think about COVID, for example, asymptomatic patients are people who are tolerant to the virus, they're infected, but they can still pass the virus on to vulnerable hosts in the community. If you're a vulnerable host, it can be really bad to be near a tolerant host if they're infected with the parasite. But tolerant hosts can also be a good thing if there's host preference. So to illustrate this example, this is data from Philornis torcans, which is found on a few different bird species in Argentina, including great keskydes, which are tolerant, and greater and little thornbirds, both of which are very vulnerable. And when you go out and survey the bird community, you'll find that when all three of these are nesting, you'll find the most parasites on great Cascades, and that's because that's where the parasite has the highest fitness. Now, when these tolerant hosts aren't available, the parasites then move on to vulnerable hosts and cause more damage. So this is really important because although this isn't the species I work with, it's in the same genus, and this illustrates the example that vulnerable hosts can actually benefit by having tolerant neighbors. If the parasite preferentially infests tolerant hosts, it can be a good thing to be around one. So this is, again, the fly I work with, the avian vampire fly, Florence downsii. And one of the host groups I work with is Darwin's finches. And unfortunately, Darwin's finches are extremely vulnerable to Florence infestation. It can kill 100% of the nestlings in a nest, and it's pretty bad. And it also has a very high prevalence in finch nests. So most of the finch nests at my field site are going to have this very damaging parasite in them. I also work with Galapagos mockingbirds, and similar to Darwin's finches, most of their nests are going to be infested with Florence downsii. But in contrast to the finches, Galapagos mockingbirds are behaviorally tolerant. They're able to offset the damage caused by parasitism, and they do this because when the nestlings are losing blood, they beg more, and so the parents feed them more, and they're able to offset the blood loss due to additional provisioning. Unfortunately, Galapagos mockingbirds start nesting earlier than finches, and they have many more flies than finches, which could amplify any reservoir effect that we see of Galapagos mockingbirds. So if we think about the players in my system, we have Galapagos mockingbirds here on the right. They could be negatively affecting Darwin's finches by increasing the parasite load on them, but they could also be maybe helping them out. If the flies prefer to infest Galapagos mockingbirds rather than Darwin's finches, it could help alleviate some of the parasite pressures that we see. Now, just how these hosts have different responses to parasitism, the parasites have different fitness depending on the host. So this is data that I collected in 2022. It's based on a lot of nests that I was monitoring in that season. And what I'm going to plot on here are the proportion of flies that pupate. So they're parasitic as larvae, and then they pupate in the nest, undergo metamorphosis, and adult flies come out. So if you have a high pupation rate, that means the fly fitness is a lot higher. And so if we look between our two host groups, this is based on over 4,000 flies and tracking if they pupated or not in the nest. We see that the fitness is much higher on Galapagos mockingbirds than Darwin's finches. And this also holds true for not only pupation rates, but the proportion of flies that are closed, so how many actually finish metamorphosis and emerge from their pupal cases. So given these large differences in fly fitness depending on host, all else being equal, these flies should prefer to infest mockingbirds. This may be a case of, okay, tolerant host may be a good thing, Galapagos mockingbirds may be a good thing for finches. So the question I'm really trying to answer is, are mockingbirds a reservoir host for Florence downsii that amplify the number of flies on Darwin's finches? And to get in more details of this, how often are flies dispersing between these two different host groups? Is it a rare event? Is it pretty common? How bad are these potential reservoirs? And also, how far are flies moving on the landscape? How bad is it to have an infested mockingbird as your neighbor? But unfortunately, they don't make GPS collars for flies. They're a little too small. So I'm having to turn to molecular methods to track where individuals go. And I'm using an analysis that's been used in mosquitoes before. It's called kin sharing to infer dispersal. And what this really relies on, if you look at this figure here on the right, it's different degrees of relatedness. So your siblings, your parents you share half your DNA with, half siblings, nieces and nephew, 25%, and so on. So you have different degrees of relatedness. And then using this information, and you can look at the spatial and temporal patterns of close kin to quantify dispersal in the previous generation. So the example I use is I was born in Minnesota, and my sister was born in Georgia 2 years later. It's like 1,000 miles away. Knowing that information, you can infer my parents dispersed that far in the time frame. I'm doing a similar thing with these flies, but I can't just ask them, hey, where are your siblings? So here's a satellite image of one of my field sites on Santa Cruz Island in the Galapagos. And in 2022, I was tracking 30 mockingbird nests, shown here in blue, and 26 finch nests, shown here in orange. And we had a total of over 4,000 pupae and larvae across all of our nests. And we had very high prevalence in the system, like I said earlier. So over 95% of the nests in my environment were infested with this parasite. It's pretty much ubiquitous. And the average number of flies in mockingbirds is about twice as high in finches. So as far as my methods go, I collected a lot of flies and I extracted DNA for whole genome sequencing using 20x coverage. And the data I'm presenting today come from 144 flies, which gives us over 20,000 pairwise comparisons for classifying how related they are. And using these genomes, I identified 120,000 SNPs after standard filtering for things like Hardy-Weinberg equilibrium, minor allele frequency, the usual stuff. And then using these SNPs, I could identify each one of these 20,000 pairs as closely related. You know, are they first degree, such as siblings, second degree, third degree, or classify them as unrelated. So now my question really becomes, are closely related flies found between mockingbirds and finches, and how far apart are closely related flies found? So if we look back to my field site, unfortunately I did not sequence all of my flies. I'm only showing data from these nests scattered across my field site. And what I'm going to do, I'm going to add lines showing when I found pairs of closely related flies between different nests. So a white line between different nests is going to indicate there's half siblings, or the equivalent second degree relatedness. Red line is going to indicate first cousins or third degree relatedness. And so when I do that, you see that there's quite a lot of lines between my different nests here, which indicates that closely related flies are not only being found between different nests, but they're found across different host species. So there's a lot of lines connecting blue circles to orange circles, so there's closely related flies both in mockingbirds and finches. And this scale may seem a bit small. The scale bar down at the bottom is only 500 meters. It's very localized transmission. But I was also looking at nests that were 10 kilometers and 20 kilometers away, trying to figure out, okay, how far are these flies moving? So in addition to these 15 nests at El Grapatero, I have sequence data from 7 nests near the Charles Darwin Research Station and 2 nests up by the landfill 20 kilometers away. And so if I again add lines with the addition of purple representing siblings, we're finding connections not only within my site, but between these different sites 10 kilometers and 20 kilometers away. And interestingly, we see a lot more connections between the station and the landfill and my field site and the landfill than we do between the station and my field site, even though they're closer in proximity. But if you remember back to the beginning part of my talk, these adult flies are not parasitic. They're feeding on decaying organic matter, so they may be hitching rides on garbage trucks or people going on a picnic and bringing their trash with them, accidentally bringing a fly back. So even though it may not be natural movement, we're not sure yet, it's still relevant for the ecology and management of this incredibly damaging parasite. So to answer the question, are mockingbirds reservoir hosts of Philornis downsii and negatively affecting Darwin's finches? Yes, it looks like 41% of all of our closely related flies found between different nests were between mockingbirds and finches, which is supportive that there's ongoing transmission from this reservoir mockingbird to Darwin's finches. And to answer how far flies are moving, closely related flies can be found anywhere from 50 meters to 20 kilometers apart. So they're traveling some great distances. As someone on my committee famously said, they're called flies for a reason. They're capable of pretty good dispersal. The adults can live for over 300 days. So they can move pretty far on the landscape. And I have a little bit of extra time, so I'm going to plug this real quick. We're thinking about, okay, why do we see fitness differences, but we don't have host preference in the system yet? Well, it's an invasive system. It hasn't really been that long since they were introduced, so maybe it hasn't evolved yet. The other thing that may be going on is the majority of these hosts are infested. So there may be some frequency-dependent selection to avoid intra-nest competition between other larvae. It may be better to go infest a finch where there's fewer ones per host. But yeah, with that, I'd like to thank all the current and former members of the Clayton Bush Lab, my supervisory committee, funding agencies, and especially Eve Galvin, who spent 4 months with me in the field chasing nests around. It was a lot of work, so I've got time for questions.